Let us now see how these monocot and dicot leaves are organized. A dicot leaf is generally known as a dorsiventral leaf because we can easily distinguish when we look at a dicot leaf an upper surface from the lower surface. The upper surface is always dark green in color, whereas the lower surface is light green in color. Hence, it is called dorsiventral. The leaf shows an epidermis, upper epidermis, lower epidermis. Between the two epidermal layers, there are parenchyma cells forming what is called mesophyll, and there is also the vascular system. The epidermis that covers the upper surface is called adaxial epidermis, and the epidermis that covers the lower surface is called abaxial epidermis. In most of the dicots, the lower epidermis generally bears more number of stomata than on the adaxial epidermis or the upper epidermis. Hence, the dicot leaf is generally designated as hypostomatic in its nature. The mesophyll that fills the space between the upper and the lower epidermis can be easily distinguished into two types as the cells are different. The cells which are very close to the upper epidermis are columnar in shape and they are closely packed. There are plenty of chloroplast and they constitute the palisade parenchyma. Whereas the cells that are present close to the lower epidermis are loosely arranged. They do have chloroplast, but they have got plenty of intercellular spaces. Hence they constitute spongy parenchyma. Thus, the dorsiventral leaf, mesophyll, is of two types, palisade and spongy. The dicot leaves exhibit reticulate venation. That's a network-like venation. If you look at a dicot leaf, you find a central midrib with the secondary veins, tertiary veins, etc. Hence, when you take a section, transosection of a dicot leaf, in the central portion, we do get the section of the midrib, and at the sides of the leaf lamina, we get the sections of the secondary veins and the tertiary veins. When you look at the section of the midrib, we see the vascular bundle surrounded by a layer of thick-walled cells forming what is called the bundle sheath cells. The entire midrib is centrally supported in dicot leaves by sclerenchyma tissue present between the bundle sheath cells and the upper epidermis as well as the lower epidermis. The epidermal hairs are present both in the upper as well as the lower epidermis, and they are multicellular in their nature. This is the characteristic feature of the dorsiventral leaf. Whereas a monocot leaf like that of a grass or maize or paddy is said to be isobilateral. The reason being that these leaves are held obliquely and both the surfaces of the leaves are exposed well to the sunlight, and these two surfaces are almost similar to look at, and hence it is called isobilateral. Besides this feature, the monocot leaves are unique in their nature as they show parallel venation, where we see the veins running from the base to the tip of the leaf. When we take a section of a monocot leaf, although like the dicot it has an upper epidermis and lower epidermis, the mesophyll tissue that is present between the two epidermal layers do not show any differentiation into palisade and spongy parenchyma. All the cells are similar. In grasses, some of the upper epidermal cells along the veins modify themselves into large, empty colorless, thin-walled cells. These are called bulliform cells. Bulliform cells are absent in dicot leaves. In the monocots, multicellular hairs are absent. Whenever there is stress of water, these bulliform cells make the leaves to curl inwards so that the loss of water due to transpiration is minimized. The leaves exhibit parallel venation, and hence when we take a section, we see the sections of lot of veins all along the leaf, lamina, with the central section being bit broader in its size, well supported by means of sclerenchyma tissue. The anatomy of the isobilateral leaf is thus almost similar to the dorsiventral leaf, except for the presence of bulliform cells, undifferentiated mesophyll, absence of multicellular hairs. The arrangement of the xylem and phloem are almost similar, but the nature of xylem vessels and xylem vessels in dicots and monocots are different, just like that of a stem.